Well, it's a pleasure to be here, and I've been to several of these, and I really always enjoyed it because the size of the audience, you get to really interact with people and have discussions. I really want to applaud the tech team. So I find this kind of room frustrating if you're using a laser pointer, because you either have to choose to like the people on that side of the room or like the people on that side of the room if you're pointing laser pointers. And so I like to write on my screen. And that was something that wasn't set up, and plugged in my computer up here and it didn't work, and so then they plugged in their Apple computer and finally got it to work, and then plugged in my computer and finally got it to work. So you guys did a great job. So now everybody will enjoy this talk more. On the talk, I'm going to really do a matrix of two things. One is going to be oral delivery and parenteral delivery, and then I'm going to focus on peptides and mRNA DNA. Of course, doing research over a long period of time requires support, so we've had tremendous support from both industrial and governmental sources, and it requires amazing students, and I have a longer list of students than even this who've contributed to this work. So here's what we're going to do. I want to tell you about our technique for making nanoparticles, which we call flash nanoprecipitation. The concept it uses is confined impinging jet mixers, and there are a couple of versions of this we'll go through that have advantages. So I'll talk about the development of the idea. And then, as I said, we'll start with oral, and this will focus on our work that is sponsored by the Gates Foundation for global health. And then we'll move over to parenteral, and we'll focus on peptides. And then sort of what our lab is now focusing on is RNA and DNA delivery. And we'll talk about, I think, the biggest challenge in that field is targeting. You know, we've all had probably most of us have had the COVID injections. That immediately goes basically to the lymph nodes and the liver, or lymph nodes that then activate the immune system, or if you do an IV, it goes immediately to the liver. And so the whole field right now is saying, how can we target to other tissues? That's really the big unmet challenge. So what motivates my group are these two quotes. This comes from a CNE News article. Over the next few years, some of the complex theranostic strategies published rampantly in chemistry journals will fall out of contention. For me, something that's too difficult to make, too complex to sustain in large-scale production is not what we're interested in. So, you know, there are lots of Science, Nature articles, and then you look at how would you make something like that, and I've been consultants with companies that had, $25 million in venture capital funding, and then after 4 months realized there was no way they could scale this up. They asked me for help, and I said, you're doomed, and they later pivoted, which is what you do in the startup world when you're crashing and it's not working, and ran out of money and went out of business. And then Scott McNeil, who is the head of the NCI Nanotechnology Laboratory. Another big hurdle in developing nanomedicines is scaling up the synthesis of the particles, developing a synthesis that yields particles on a consistent basis. That's still a difficult process, and that, I think, is what we have been able to do. So this nanoparticle world is really schizophrenic, because the group that wants oral drugs, as was presented by the previous speaker, with really an excellent talk, are trying to get things to be hydrophobic things or things that aren't bioavailable, trying to get them more bioavailable. And so they want things to sort of immediately dissolve. So here's some of our work showing a drug which is essentially insoluble, and you put it in our nanoparticle form and you get substantial release. So that's one community. They want things to be put in form and then immediately be released when you take it orally. But the other community is the parenteral community, for example, COVID vaccines, where you don't want things to be released immediately. You want them to be captured and carried with your nanoparticle to the site where it needs to be delivered. And so those two things are completely orthogonal to each other. I want it to really dissolve quickly, or I want it to be captured and not dissolved until I get to the site of interest. And this is some of the work we'll talk about there. So what did we develop? In the last, it's been over 15 years, we developed this technology, which we call flash nanoprecipitation. And if you're trying to do a precipitation and you want to control size, you need to do two things. You need to have everything very supersaturated, and you need everything to be supersaturated exactly at the same time. Okay, so everything has to be supersaturated, and everything has to start at time equals 0 to control precipitation. And then you have to stop the growth at some later time. So that's the key. Well, the way we start the precipitations and the way we get homogeneous precipitation is to use these confined impinging jet mixers. And what that means is we're going to take our solvent phase, which in what I'm showing you here would be a stabilizing polymer, for example, a blockopolymer with a hydrophobic block and a polyethylene oxide block, for example. And then the solute, our drug molecule there, in a water-miscible solvent. So if you're all familiar with COVID vaccines, that would be the lipid and then the corresponding cholesterol and other phospholipid components, and the stabilizing PEG lipid would go in here. Now, later we'll talk about mRNA. So in that case, the mRNA would come in the non-solvent, the aqueous phase. But what we're going to talk about first is putting all of the components in this water-miscible solvent. So they're coming in there. Our anti-solvent is going to be water. So now what we do is these two jets are aimed at each other, and they cause intense turbulence. Turbulence is swirling flow, and it winds up those two fluids to end up with very thin layers between the solvent and anti-solvent. So let's say ethanol water phase. And you use a turbulent intensity so that the thickness of those striations, what's a striation? Well, if you put cream in coffee and you're stirring it, the white stripe of cream gets thinner and thinner as it whirls around. So that's what turbulence is doing, is spinning these layers to make these striations or thicknesses between the solvent and anti-solvent. Gets down to about a micron in 1.5 milliseconds. And so that diffusion time is then about 1.5 milliseconds for the solvent anti-solvent. And that's crucial because you can scale this with turbulence, but you can't scale these precipitations in a big vessel because you can't get things mixed uniformly to go from dissolved to supersaturated in a large vessel in that kind of time frame. Okay, so we're using turbulence in these confined impinging jet mixers. I've shown coming out here schematically, I've separated the stabilizing polymer from all the core components from our drug, but of course they're mixed in space. Then what's happening, you needed that really high supersaturation so that things stick to the first thing they see. And that allows you to do drug cocktails and combinations of different things because everything is supersaturated. Everything wants to stick to the first thing it sees. It wants to get out of the aqueous phase, the anti-solvent phase now. And they start aggregating and growing. So the hydrophobic core starts growing. The hydrophobic blocks of the polymer start growing on the surface of that, aggregating with that, until you build up a dense enough stabilizing layer to stop particle-particle aggregation. So the start process is mixing. The stop process is having a thick enough layer that it stops particle-particle aggregation. Okay, so that's the controlling the stop time is by controlling the formation of that layer. Now, what's shown here is how do we control particle size, and this is particle size. This is Reynolds number, which is basically the jet velocity going in the mixer. And if you do the mixing too slowly, you end up with large particles and aggregates. If you do the mixing rapidly enough, then importantly what happens is the particle size becomes independent of the mixing. What that means is the solvent anti-solvent switch has been faster than the time scale for that diffusion limited aggregation. And it doesn't matter how much faster you go, you get the same particle. This is a real advantage in processing because unlike maybe an emulsification process where if you put in more energy you make smaller droplets, here we make the same size particle regardless of changes in flow rate. And that's a big deal if you want to make different volumes of material. It's one of the reasons the Gates Foundation approached us about our technology, because you can scale it in time rather than scaling it in intensity of mixing and changing particle size. Now what's shown here is the other way we control particle size is by changing supersaturation I mentioned. So for example, if I take a condition with high supersaturation, then I have more nuclei forming. The drug accumulates on a greater number of nuclei, so the average size is smaller. If I have less supersaturation, I have fewer nuclei, so the drug accumulates on a fewer number of nuclei, so the average size is bigger. So we've changed here from 200 nanometers down to 65 nanometers by changing supersaturation, okay? So those are the two main handles to control nanoparticle size. Now, I call these things confined impinging jet mixers. There are a number of ways or geometries you can use, and I'll show you two that we use widely and that are used industrially widely. This is the two-jet mixer. That was the cartoon I just showed you where the solvent's coming in one stream, the anti-solvent another. That's a computational fluid mechanics model of this, and it's showing you that all the energy is going into the center of that chamber, and that's where all the mixing occurs in that high intensity energy dissipation zone. Everything goes through that zone. Now, this second geometry here is we call a multi-inlet vortex mixer. That is, we have 4 inlets here. The flow swirls around and is turbulent, and then exits the bottom of that. But once again, the highest energy is in the center, and this is important because, in fact, you prevent deposition on the walls. And this is the reason that microfluidics will never be scalable. Okay, the COVID vaccines, next slide. So all the COVID vaccines are made using our confined impinging jet mixers. So the president, I was really quite surprised, Mike McDermott, who's the president of Pfizer Global Supply, took CNN through all the Pfizer manufacturing process, which most of the time pharmaceutical companies are highly secretive of how they do things. As the previous speaker said, I'm not going to tell you quite what our solvent mixtures are, which is fair enough. But Pfizer took the press through their production facilities, and the comment, and this is their confined impinging jet mixer, so the anti-solvent, solvent come in here. This was their computer animation of what that looks like in the precipitation, and his comment is the impinging jet mixer makes it possible. So it makes it possible because, again, the mixing in the center, you don't get deposition on the walls. If you try this doing with microfluidics, there is always either the alcohol stream with all the lipids or the aqueous stream with all the mRNA is in contact with a wall. You'll get deposition on the wall, and it will slough off. And sloughing off is just like if you're in the antibody production world. The denatured protein gives you an immune response and is very bad. So those sloughed off mRNA aggregates give you a huge immune response, and that's why it's not scalable. Now, if you just make a small run to get a lab sample, you can use a Precision Nanosystems, nanosampler, it's fine, because you're not running it long enough for that wall deposition to occur for things to slough off. But that's why all the major players use confined impinging jets to make large-scale things like the COVID vaccines. This just shows the particle sizes that are easy to get. So here we've made 450 nanometer particles. There's 6 times more drug than stabilizing polymer. The data I'll show you with the Gates Foundation, we make nanoparticles that are 90% drug. So we make these really, really highly loaded particles in this process. We change supersaturation. We make, in this case, 88 nanometer particles. Here were a few empty micelles that we made. The important part here is that everything is below 0.2 microns. So you can put this through a 0.22 micron filter and make it sterile, which is just crucial, for example, for the COVID vaccines. If you try milling down, you'll always end up with some log normal tail that will blind filtration. So you can never mill down. The average size may be below 200 nanometers, but you'll never mill down to a size that you can put through a 0.22 micron filter for sterilization. Okay, so we'll start off with the oral drugs. The Gates Foundation approached us about 6 years ago now and said they thought our process was the one that had the potential to take some of these very poorly bioavailable global health drugs and make them in a more bioavailable form. So we've done these 6 and more drugs with the Gates Foundation, and they've all been successful. Lumefantrine is the one that they have paid for WuXi AppTec in Shanghai to put together a GMP line, and so that's in place for anybody to use on a contract basis. Okay, so this is now, we make it in our lab, milligram scale, and WuXi makes it in kilogram scale. Okay, so lumefantrine, it's an anti-malarial drug, and its problem is, if you just look at this structure, it's really hydrophobic. It has a log P of 9. And so it has 2 pharmaceutical challenges. The first is bioavailability is dependent on food intake. So what's shown here is the sort of feasted state release, and along that axis is the fasted state release. So there's a huge difference. And this is a big deal for the Gates Foundation because they're very interested in pediatric therapeutics. And if you have a child or an infant with malaria, you don't know whether they have recently had diarrhea or thrown up, so you have no idea what their stomach state is, and therefore whether they will, this will be bioavailable as a fed state or fasted state. In fact, there's a 19-fold difference in the bioavailability between fed and fasted state with lumefantrine. So that's first challenge. The second challenge is we had discussions about spray dry dispersions, which is a very powerful technique for making drugs. However, in the Gates world, you need to have stability at 50 degrees C, 75% relative humidity. And so there was a major pharma company that made a spray dry dispersion of this, lumefantrine. Here was the drug release percent at time equals 0, but after a few days, the release dropped by 50%. So this dramatic change in bioavailability is just unacceptable, in the global health setting. Now, you can get around it. Why does this happen? It happens because at the high humidity, the matrix material gets plasticized, the drug can diffuse, gets kicked off the polymer, diffuses and crystallizes. You get around this if you put it in blister packs and kept it away from moisture, but the Gates Foundation said, no, that's way too expensive for global health. It has to be a bottle that's sitting on someone's shelf and you can't guarantee whether they've closed it or not. Okay, so these are the two challenges with lumefantrine. The other challenge is, in the other things we've talked about, we're using beautiful block copolymers, PEG PCL or PEG lipids for the COVID vaccines. Those are way too expensive for global health. And so we have looked at a number of stabilizers that are really cheap. So HPMCAS is one of them. That's relatively inexpensive. Lecithin, which is PAM, you spray on your frying pans to keep things from sticking. And Zine, which is a really interesting protein from corn, which is a lysosomal storage protein in corn. And you can make all of those work as stabilizers. Interestingly, for those of you who are from the pharmaceutical world, the reason that the Gates monitoring group decided to go with HPMCAS, even though Zine was slightly better in the animal test, was because Zine is a natural product and it changes depending on when the corn was harvested and how wet it was, how robust the corn was. And so it was the lack of reliability in that, even though we eat it. It's grass. But in terms of having the FDA approve it, HPMCAS is a synthesized material, and you know what its characteristics are. That was a surprise to me as an academic. That's something, you can eat corn, but that's not, there are real problems with getting through the FDA approval process. Well, so we worked on this, and what's shown here, so there's a large paper that goes through all these details. This is our very small scale, the 2-jet mixer in our lab that we work with sub-milligram samples. This is the MIVM, the next scale up. This is the larger scale, which is 5 liters per minute that we built, tested, and sent to Wuxi. And the really important thing is here are the nanoparticles made by the small scale mixer, by this medium-sized mixer using syringe pumps, a medium-sized mixer using continuous flow. These happen to be Coriolis flow meter control pumps that we used, and this is at Wuxi Aptec. So what you make at the bench, scales, and that's really crucial in the nanoparticle world because many techniques don't scale. And as I said, these nanoparticles are 90% drug, which really allows you to deal with these low bioavailability drugs. Now here are the really striking results that make this interesting. So this is release kinetics for these. So the way we make these, we make the powders, and then we spray dry them through a Buki 290 in our lab, a small lab in Wuxi. They do much bigger spray dryers with some cellulosic excipient to sort of stabilize these powders. And so these are the dry powders that have been then aged 50 degrees C, 75 relative humidity. This is the original, going 4 weeks under these accelerated storage conditions. This is release under fasted conditions and under feasted conditions. We have a lot of lipids in the release media. And what you see is, first of all, the release is identical between fasted and fed state conditions. And that's really quite surprising and quite helpful because it gets around this problem of whether the infant has a full stomach or an empty stomach. Okay, why does that occur? It occurs because the release is not dictated by the solubility of a lumefantrine crystal, but rather by the amorphous material we've trapped in these nanoparticles. Okay, so that controls the release. Independent of what's out here, it's the release out of the particle caused by from the dissolution of that initial 100 nanometer amorphous core. Now, the other surprising thing is stability. So both under these aged at 4 weeks under these conditions, you get the same release kinetics. Why does that work? Well, it turns out in this paper, which is the previous slide, it turns out that the drugs are primarily amorphous, but have some nanocrystalline domains, and that's seen by solid-state NMR. If you put these XRD, you don't see any crystallization. Solid-state NMR is the really way to look at aggregation state inside 100 nanometer particles. And our collaborator there, Aditya Rawal from University of New South Wales in Australia, shows that what you have are in this 100 nanometer core, you have about 30% of crystals that are less than 10 nanometers in size. And it's those crystals that stabilize the amorphous core so you don't get increased crystallization when these are exposed to humid conditions. So you end up with something which is really dead stable as opposed to the spray-dry dispersions where the moisture kicks the drug off the polymer, it diffuses and crystallizes. Okay, so that's one story. So this works really well for global health and taking these very hydrophobic materials and making them into nanoparticle form. Now let's go over to parenteral administration, and we'll start off with what do you do when a particle is not quite hydrophobic enough to make a stable nanoparticle. If it's not hydrophobic enough, you may do the precipitation, but then you get a process called Oswald ripening where there's too much solubility and it'll go and find other nanoparticles, and they grow with time fairly quickly. Well, what you can do is to ion pair. So this is a drug, an antihistamine. You ion pair with pomoic acid, so they come in a common solvent, they precipitate, they form the ion pair and precipitate out. So in the pharmaceutical world, this would be salt form engineering, okay? Except we're doing salt form engineering at a 100 nanometer scale. And so now you can easily make these 100 nanometer particles which are stable indefinitely from this material which is too unstable to give you nanoparticles. It's too hydrophilic to give you stable nanoparticles over time. And that gets us, now let's go from that small molecule, we've done a whole bunch of those, let's go to the next bigger molecule where we do ion pairing. And this was a problem brought to us by BMS. And what they had, they're interested in peptides, and they have lots of peptides, and they just were interested in why these things are not bioavailable and what they can do to make them bioavailable. Would nanoparticle formulations help? And so they gave us a whole class of peptides. All of them had less than 0.1% bioavailability in their screens, okay? So these are things that, are not therapeutically interesting because they're just so non-bioavailable. And I'll focus on, so they gave us 4 of them to look at, I'll focus on 1 and 3. And so this first one, which has a few more acid groups than amine groups, is so tightly hydrogen bond and coupled that it's insoluble. So it's not bioavailable because it doesn't dissolve. Okay, so it's over in this BSC class of things that are, don't get through membranes because they're not soluble. This one with 8 carboxylic acids is super soluble. It is not bioavailable because it can't get through the GI tract. Okay, it's too soluble to go through the cell membranes at the surface and to be active. So they fail for completely different reasons. So what we're going to use is the 4-jet mixer because we bring in the peptide and DMSO, we bring in our ion pairing agent and the block copolymer stabilizer in another mixed solvent, and then we have 2 streams here which are anti-solvent stream. So it's a more complicated formulation issue. You need different streams because things are soluble in different streams. You can't put the peptide in with the ion pair agent in this case because it precipitates out, so they're not equally soluble in the common solvents. You need them in different streams. And we can make these particles. So here are the results. So we'll take that first one, which I said is insoluble, and we'll ion pair it with a dodecyl sulfate, simple SDS. What's happening now is that you shift the pH down and turn off the carboxylic acid charges. The sulfate stays charged, and the amines are charged. So now you can make these 100 nanometer particles. They precipitate really well and they're stable. And you've now taken something which is insoluble. This is the release assay. This is time in hours. Insoluble, and you made it very soluble, okay? Because now the SDS comes out and it releases the peptide and you've made it soluble. The second one is the one that was way too soluble. Well, again, we're going to take a sulfate. We'll shift the pH down. These become charged. This gets turned off, so we have now something cationic. It will complex with the sulfate, which stays charged even at pH below 4. The cholesterol group is hydrophobic here, and now we also include in that another hydrophobic component, because what we're doing is now making a matrix, a hydrophobic matrix, to include that peptide so it doesn't escape. Again, you can make these nice particles, and now we take something that escapes way too easily, and we made it into a nanoparticle form, which stays encapsulated. So there's a lot here you can do with these ion pairing recipes to either make things more soluble or less soluble, depending on what your therapeutic goal is. Let's go now, we'll focus on the work we're now doing, which started about 15 years ago with Merck, and that was siRNA. So they had just bought a company called Sirna. They spent $500 million. It was the biggest project inside Merck. They thought they were going to control siRNA delivery, and they realized they had no idea how to scale up the process. They approached us and said, would our technique work? And we had no idea if it would work, because at that point, the cationic lipid and the stabilizing PEG lipid comes in an ethanol stream. The siRNA came in a buffer stream. For our technique to work, the electrostatics have to be faster than the particle assembly. That is, the cationic lipid has to bind on the siRNA, make it hydrophobic, so it starts precipitating, so then the PEG lipid will stabilize the surface. But it works beautifully, and we made these 60 nanometer particles with Merck. The sad part of this story, we have a beautiful patent. Merck abandoned siRNA. They got out of the field, and so they abandoned the patent, which would have covered all the COVID vaccines, interestingly. And that's a sad kind of story. They funded the student, Varun Kumar, in my lab. Of course, when a company has put 4 years worth of money into a grad student, it's great if the student goes to work with a company, and Varun said he didn't want to work with a big company, so he went with the Nylum, which was a small startup at that point, had a couple of hundred people. And when Merck got out of siRNA, they sold all their intellectual property to Nylum. So here Varun has his patent and everything's looking great. And then Nylum gets sued by Tecmara. So if you're in this siRNA, mRNA field, everyone is suing everyone. This company, Tecmara, sued Nylum. Nylum lost. And part of that suit, they could not make lipid nanoparticles. So they could continue with things they had in their pipeline, but they couldn't do any new lipid nanoparticle work. So Varun at that point, and now so everything they do is stitched siRNA. So Varun felt, crestfallen that now he couldn't do anything he was really good at. So he left and was an investment banker and is doing really well. And of course now, Nylum, they didn't continue the patent. So it was abandoned because Nylum had no interest in doing lipid nanoparticles. So that's why both through Merck and then Nylum got abandoned. Okay, so now let's look at mRNA and DNA. It's clear that we can scale up RNA, right? Well, COVID vaccine. So that's really simple. Turns out that the next harder one is going to be DNA. It's a lot harder to encapsulate. It's a lot harder to work with. And so any of these formulations involve an ionizable lipid, and then a neutral zwitterionic lipid, and then the PEG lipid is a stabilizer, and then cholesterol is added. Each of those have a different function. And we'll just show you we could either make this in our 2-jet mixer or a 4-jet mixer. Now, why DNA is more challenging? siRNA is easy because it's so small and so anionic. DNA is much bigger, so it's harder to capture. And also, in its biology, if we have an mRNA particle, it just needs to go into an endosome, get out of an endosome, and then it's active in the cytosol, and it builds the protein, the spike protein. For DNA, it's got to go from here, the cytosol, into the nucleus in order for it to replicate. So you've got another set of barriers to go through. So the DNA delivery in biology is much more complicated than mRNA delivery in biology. And so we're going to look at different ways of making these nanoparticles with our technique. So the first thing is, whether you use the 2-jet mixer or the 4-jet mixer, you make exactly the same particle. So again, that's the scalability and an advantage here of using a turbulence. It's not the outer geometry, it's the turbulence intensity that controls the mixing and the precipitation. And then I showed you earlier a slide that showed mixing is poor, big particles, you get it fast enough so it's the mixing diffusion of the anti-solvent solvent is quicker than the assembly, and then the particle size is constant. That's exactly what we see here. So this is a series of increasing flow rates using a syringe pump here, and you get to a high enough flow rate, and the size is independent of the flow rate, making 60 nanometer sizes. And this is using a handheld, a really small version. You can make exactly the same particle with handheld. You don't have to go with all the volume and complexity of syringe pumps. Now, the big challenge in this field, both the mRNA field and the DNA field, is targeting, getting it to other tissues. And if you look at what people are trying to do in targeting, so this is from a really beautiful study. I'm saying nothing negative about the study. This is a group of good friends at UPenn. But the way they put a targeting ligand on the nanoparticle is they take the antibody and they conjugate it with a hydrophobic tail. And then they're relying upon this, which was actually liposomes, but they're using the same thing on the lipid nanoparticles. And they put this, which assembles into a mice cell, in with the hydrophobic LNP. The antibody pulls out of that mice cell and deposits on the surface of the LNP. And it works great. You can load it up with antibodies. The problem is, how did it get there? It got there by being dragged out of solution, because the antibody is so big compared to the lipid tail you've got. So if it comes on that way, it's also going to leave that way. So if you're familiar with the COVID vaccines, with their PEG lipid, that PEG lipid partitions off in 30 minutes. It doesn't stay on the surface. So anything you try to do to make it targeted is going to pull it off even faster. So none of the, in my opinion, none of the targeting studies, and I'll be glad to talk to people about it, are really going in a direction which is going to give you a solution to this targeting of LNP's DNA, mRNA. So what we're doing, so this is just a picture saying, if you take this traditional approach of a PEG lipid on antibody, it's going to pull out. And what we're doing is taking our antibody PEG on a PCL block, a big polymer block. So it's much more hydrophobic. It stays anchored irreversibly in that nanoparticle. So now that it's irreversibly anchored, it can target, it can act like an antibody. Now the big question, and we're starting, and I'm giving you kind of where we are on this, and in a couple of months we'll have more results, but I'll tell you sort of our intellectual trajectory here, and that is, but you have all this PEG here, which is irreversibly bound. Is that going to screw up the endosomal escape? If you have too dense a PEG layer, then that nanoparticle may get internalized in the endosome, but it may not get out of the endosome. If it doesn't get out, then it's not effective. What we've recently been able to show, this really talented student, is either on a HeLa cell line, which represents a phagocytotic cell line that wants to take things up, or a JERCAT, which is a T cell type line, that we get better uptake and transfection of DNA than you get with the current DMG PEGs, and on either of these cell lines. So that's really neat. It says the PEG is not going to screw up the internalization, getting out of the endosome, getting into the nucleus, and therefore you can put a few targeting antibodies on it and it's not going to mess that up. So this is where we are now. The JERCAT cell line, if you all know of CAR T therapy, where you take out antibodies, cancer cells from a person or antibodies from a person, and you grow them up and then re-inject them. Novartis was the first one with that in the field. It's $450,000 for a treatment, and that's just, our medical system is not going to be able to tolerate that. But if you can do it endogenously, if you can inject a nanoparticle, which then gets into circulating T cells and reprograms them inside the person, you really have a chance of having a therapy that makes sense. So that's where we are. I've just told you about our mixing technology. We've gone through some of our Gates work and very hydrophobic drugs. You can make them much more bioavailable. You can do this with peptides and proteins. You can do it with RNA and DNA. And we think the next sort of barrier and challenge is going to be targeting that we're working on. In a couple of months, we should have the first results on the targeted materials. We're working with a group at UPenn on that research. So I'm glad to answer any questions.
Thank you both.
Okay.
We got time for a couple of questions here just before we move on. Shaukat?
Bob, actually, I like your presentation. You have sent the paper, actually, lumefantrine, actually, this is very exciting, actually, molecule. My question to you, when the Gates Foundation actually wanted to develop just one drug, because if you look at the Cortem, approved by Novartis, they have two drugs, right? So it's a fixed dose combination that is about 20 milligrams and other is 200 milligrams, right? Why not you develop this glufantrine with the artemether? I mean, because this is the real drug, actually. I don't know whether glufantrine has been approved for the anti-malarial drug. I'm not sure. But I think the CORTEM together has been approved. That's one question. The other question is actually on ion pair. I know you talk about the ion pair. When you're making the ion pair, are you also making the ionic liquid? Or how do you really, can you really also make the ionic liquid and then making the nanosphere from the ionic liquid composition?
Okay, say that, the last part again. Can I?
The ionic liquid. Right?
Ionic liquids?
So, yeah, so combining the positive charge with the negative, right?
Yep,
That's what we're trying to do. And there are some publications actually from groups looking at the ionic liquid, you know, which really basically making a salt and then making actually emulsifying with a certain surfactants to, better absorb, have you tried, making, ionic liquid?
Yeah, it turns out those components that are in the ionic liquids, which are really spectacular, GI enhancement materials, are just too soluble to be put in nanoparticle form. Each individually will come out, even though you can make an emulsion, over a time scale of a nanoparticle, they come apart. They're not quite hydrophobic enough. So we have looked at that. Your first question is, yes, right now in the third world, lumefantrine and artemisium together are used in 2 different tablets. And you can't make them into one component in our technique because their solubility is very different. Lumefantrine is really hydrophobic, artemisinin is not that hydrophobic, so people are given them individually. Now what we've shown, I didn't show you the animal data on that, so we got 4 times the bioavailability of the lumefantrine, which is now given, and right now it's given as 4 tablets over a 3-day period, and so compliance is a real problem. So it looks like with our formulation we can get down to 1 tablet a day, which has positive compliance implications.
Yeah, I have a couple of questions. One is about your collaboration with BMS and the peptides. So you showed data in which there was one peptide which had extremely low bioavailability because of low permeation through the intestine, right? So you showed data of how you reduced the solubility in the percentage release, but how did that impact the permeation? Yeah,
so then what you had to do is add permeation cap rates, permeation enhancers with the nanoparticles to get enhanced bioavailability.
Okay. The second question is in the lumefantrine work, you said that there was some stabilization because of maybe 10% of the drug had crystallized in those particles. But wouldn't that actually result in greater crystallization because then they'll act as seeds, either in the solid state or during dissolution, so you cause precipitation?
Yeah, I agree. The stability is quite interesting and amazing. And initially, we thought it was completely amorphous. But then it was a solid-state NMR said, no, you actually have these little crystallites that must be so defective that they prevent crystal-crystal motion and fusion. And they appear to prevent scavenging of the remaining lumefantrine into those crystals over time. So it is stable. But we completely agree. It's unexpected, that degree of stability on something which has some crystallinity in it. But it's the fact that it's 10 nanometers. That's why it doesn't show up in XRD. It's not big enough or cooperative enough to give you a good XRD signal.
Okay, let's give Bob another round of applause. Thank you very much. Thank you. Bye.